Okay, welcome everybody to the August MAC. We're going to go ahead and get started. I'll introduce the speaker. Our August speakers had a highly unusual and interdisciplinary career trajectory. He graduated with a bachelor's in mathematics from the University of Otago in 1978, and then took a job at the University of Auckland as a programmer, biometrician, finished a PhD in zoology and neuroscience from the University of Auckland, then 2 postdocs in Los Angeles at USC and Caltech. Also joined the biological computing group at the Jet Propulsion Laboratory in NASA. In between all that, 33 years as associate professor of zoology at the University of Otago, and he just retired at the end of last semester. So I figured as long as he's working for free, he might as well be working for us for the next 40 minutes or so. Please give a warm Mark's welcome to Associate Professor Michael Pollan.
Thank you, Travis. Can you hear me? Hello.
Go.
Can you hear me?
Yes.
Okay. All right. Thanks for that introduction, Travis. It's true, I have just retired from the university. Can I share my screen here? Where are we? There. How's that?
We can see it.
Okay, so I've just retired from the university, but still interested in the same old questions and still working on them. So I'm really grateful for this opportunity to tell you about things that I've been playing around with. As Travis said, I was actually an undergraduate at Otago University in the 70s. And in those days, we would take the summer off, and I went with my girlfriend to Australia in 1976, and we ended up camping in the Flinders Ranges in the summer of 1976. I was a mathematics undergraduate in those days, and if you told me that I'd be back there 40 years later with the same girl still driving a Holden, I might have believed that, but I would never have believed that the reason I came back was because I thought that the secret to understanding how brains work was hidden under a rock behind this pub. But it's true. Now, most of this talk is actually going to be about the evolution of digestion and feeding. And I kind of apologize for that because I know you're interested in brains, but it turns out, I think, that brain evolution is basically about the evolution of the gut. And once you have guts, brains are really easy. So let's have a look at here's the Blindman pub in South Australia. Let's have a look at the rocks and behind the pub. Something weird going on there. All right, so what you're looking at there is the remains of an ancient seafloor that's been kind of scrunched up. And if we zoom in on a slightly unscrunched up bit of it, there is the Burkina Gorge World Heritage Site. So Blindman is up there. That's where the pub is. We're about 25 kilometers south. Here's a slice through the ancient seafloor. Over on the side, so it's a corner of an ancient seafloor that's being tipped up and it's sticking out of the desert in South Australia, and that's the Flinders Ranges. And this surface over here is the floor of a shallow ocean from the Cambrian time, which is interesting because that's when all modern animals appear in the fossil record. In fact, in Darwin's time, this is when any animals appeared in the fossil record. And in Darwin's time, there was no fossil record of animals preceding the Cambrian, and that was a bit of a problem. But if we look over here, this kind of light bit here is a material called tillite. That's material that's been dropped off a glacier. So that's the end of snowball Earth. And just as the Earth started to warm up, we get the start of this Ediacaran period, which is named after this region in South Australia. A new geological period created in 2004. So, so not surprising, apart from the fact that I was a mathematician, that Helen and I knew nothing about the fossils that were here at this time, because these fossils were only just being discovered last century, and this era has only been just defined in 2004. So across here, there's 8 kilometers representing sediments laid down over 100 million years before the Cambrian explosion. So that contains fossils of animal history preceding the Cambrian explosion, preceding the appearance of modern animals. So let's take a look and see what was there 500 million years ago. On the floor of the shallow ocean, we see lots of very flat animals that look like this. We're very confident these days that these things are animals. This is one called Dickinsonia. You can see its feeding traces out behind it. And because there's nothing on land, every time there's a big rainstorm, they got buried in sand and mud. And there's a very good fossil record of these things these days. So here's the fossil that I modeled that off. And that fossil's taken off a rock that formed the entire PhD of the student from the University of Adelaide. There's something like 100 different animals on this one, fossils on this one piece of rock. There's a lot of animal fossils 500 million years ago in Ediacaran. To figure out what kind of animal it is, go back even further. We'll go back another, 20 million years. Now we're in the deep ocean in what is now the Atlantic. So this is was around the equator in between what is now America and Africa, called Apollonia. We dive down deep. We find these creatures that kind of slightly resemble a Dickinsonia. And there's been a lot of debate about what these things are, but so this thing is a few centimeters across. It can't be a plant because it's 2 kilometers below the surface of the ocean. So what is it exactly? It's an animal called Fractifusus. It has a very unusual body plan. It's not like anything that we see on Earth today. And in particular, it's got segments, but the segments are offset. So it has segments like an animal, but the segments are offset on each side. It's what we call glide symmetry. So here's a quick look at the phylogeny of animals. I've just put in major groups of animals up here. Now, when I'm trying to put this together, there's basically no way that you can put onto a diagram like this everything we know about these animals. It's just, there's no way that you can make everything make sense. And I think that's why people are really keen on molecular phylogenomics, on molecular techniques for doing phylogeny, because then you've got an algorithm, right? You've got something that'll tell you what the right answer is, even if it doesn't make sense, but everybody can agree on that. Molecular phylogenetics has figured out the relationships between pretty much every animal on Earth these days, with the exception of these things, the ctenophores. These things are weird, right? So philosophers always ask the question of what, if we found life on another planet, what would it look like? Well, this looks like something from another planet. This is something else. So we'll just set it aside for now. We can't get the molecular evidence for these Ediacaran fossils, but on morphological grounds, we can place them in relationships to these other things. And there's some evidence that the ctenophores are related to these very old rangiomorph animals. This is us, the vertebrates up here. So everything up here, which is alive today, turns up in the rock record in the Cambrian, and everything that's down here, the things that we find in Ediacaran and lots of other places around the world these days, are gone. They disappear from the fossil record at this point. But the molecular clocks tell us, if we look at the rates of molecular divergence in the genome, the molecular evidence tells us that we were all there from the beginning. So all of these major groups of animals should be in the Ediacaran. The molecular evidence says, and I would have said 10 years ago, the molecular people are wrong, but the signal keeps coming back time and time again. And at a certain point, you got to start taking it seriously. We should have been there, but there's nothing here that looks like us. So there's nothing in the Ediacaran that looks like a modern animal, with some exceptions, yes. So, well, we'll see. These rangiomorphs and Dickinsoniomorphs have segmentation, so they are divided up into segments like animals. They grow like animals. This is another reason that we think they're animals, is they don't grow like plants. Plants grow by sprouting new bits off at the tip. Animals grow by expanding and dividing up as they grow, and these things clearly grow like animals. So we think they're animals. And there's evidence now that they're animals for the same reason that we're animals. They have the same genes that set up that segmentation process as we had. We'll look at that in a minute. So the evidence says that the genetic machinery for making animals like us is very ancient, that it precedes the very first animals in the fossil record, and yet animals like us are not there. And we don't see this glide symmetry, which is almost ubiquitous in the Ediacaran. We don't see that in modern phyla, right? So here's just a snip out of that little movie. Here's Fractophusis. Here's another vertical animal called Charnia, all exhibiting this kind of offset glide symmetry. So the segments on each side are slightly offset from each other, 90 degrees phase offset from each other. Trust me, we'll get to brains soon. So we can use molecular techniques and anatomical techniques to ask, who were these guys and how are they related to us? It's really unclear. Another approach we can take is the ecological approach. We can say, well, who lives there? What kind of animal could live on the ocean floor, on the abyssal plain, 2 kilometers or more below the surface of the ocean? So we go looking there today, see what kind of animals live. Ooh, you're not seeing that, are you? Hang on, I'll just see. I do not know why that is not showing. We'll just yeah, we'll just carry on. So use your vivid imaginations, and I will explain. So what lives there? I'll see if I can. This is actually kind of a cool movie. We don't have time to muck around here. What lives at that depth today is echinoderms. So this is the realm of echinoderms. And those are things like starfish and sea urchins are the familiar ones, brittle stars. There are some called crinoids, and the crinoids are the really interesting ones. The crinoids are these filter feeding animals that sit on the ocean floor that are sometimes called sea lilies or feather stars. And they are bottom feeding filter feeders or detritivores. I'll see if I can get that movie, because these things are echinoderms basically live off marine snow, which is organic material that coagulates and precipitates through the ocean. And it's there's a lot of organic material in them, but it's very difficult for bacteria to digest, or for single-celled organisms to digest it, because it's packaged in polymers that are very difficult to digest. So I'm sorry we can't look at that, but another thing that is in that video that I'll see if I can get forwarded to you, another feature of echinoderms is that they have internal skeletons. So we talk about invertebrates, and we say that echinoderms are invertebrates because they don't have backbones, but in fact they have backbones. They've got backbones in every arm, right? So they have vertebral columns in every arm, internal skeletons like us. So it's a bit unfair to call them invertebrates because they actually have more backbones than us. So here's some things that you would have seen live. These are animals. Here's a sea lily or a crinoid. Here's another animal called a sea pen. Right. So I said on the earlier slide that kind of glide symmetry is never seen in modern animals. Well, in fact, it is seen in modern animals, and I'm slightly puzzled as to why everybody says it's not, because it is. It's not seen as a phylotypic characteristic. Because the echinoderms have pentaradial symmetries. The echinoderms have 5 of these branches, but each individual branch has exactly the symmetry that we see in these Ediacaran creatures, and not just, and you can see here's Charnia, this leaf-like thing that lived on the seafloor in the Ediacaran, has the same kind of overall morphology. It has the same kind of primary, secondary, tertiary branching at right angles with this offset. See this little 90-degree phase offset on the branches here. So exactly the same, right down to the level of 1, 2, 3 levels of primary, secondary, tertiary branching we see in Charnia, and the same thing's going on in the sea pens, right? So we do see that symmetry in modern animals, and it's adaptive for these creatures because they're all feeding on this detritus, on this gunk that floats down in the ocean, the marine snow. And that symmetry works for them because what they're doing is they're passing these food particles by cilia. So they've got these waves of cilia that are passing the food particles down the tertiary branches, along grooves on the secondary branches, down grooves on the primary branches to the mouth and into the gut. And that offset symmetry means that those waves of cilia, so they're basically sweeping the food particles down into the mouth, into the gut. Those little sweeping processes are not fighting each other, right? So if you had bilateral symmetry here, then the cilia sweeping the food down here would be fighting the cilia sweeping the food in the opposite direction. So that offset symmetry makes this feeding mode more efficient. So it's adaptive for these animals. And what they're doing is they're taking those food particles which with single-celled organisms, bacteria, and single-celled eukaryotes can't digest, putting them into a bioreactor, this acidic solution with bacteria in it that can, with the aid of the acid and the enzymes released into that stomach, they can digest that material, and it comes back up the inside. The inside of these things is hollow, and that nutritious liquid then flows back up the inside of these tubes, right? So that's the feeding mode of echinoderms, and very similar to the feeding mode of the sea pens. And if we look at that feeding mode in modern animals, it's just these couple of weirdos, right? In amongst all of these animals, there's these 2 weirdos that feed that way. They're distantly related. It's regarded as a kind of non-interesting thing because it's not a symmetry that you see in the other animals in those groups. It's independently evolved, and it's a specialization for that particular feeding mode. That's what they say. So here, in fact, is a sea lily feeding. If you watch this food particle here, it's got the sticky cilia. See, it's sweeping that food particle back down towards the mouth. You can see that one, right?
Yep.
Okay, so the modern phyla with glide symmetry have that symmetry because they are sweeping food particles along channels to get into these bioreactors with the bacteria for extracellular digestion. That gives them access to a resource that there's a lot of nutrition in that resource floating down through the ocean that other organisms can't get at. And so the suggestion is that the rangiomorphs that have that same morphology occupy the same or similar niche, and the Dickinsoniomorphs, the assumption would be that they have a similar kind of feeding mode. The Dickinsoniomorphs are not sitting in the water, kind of sweeping the food particles down into the mouth. They're flipped upside down, and they're crawling over the surface, sweeping food along these channels, doing the same kind of deal, sweeping food along channels into some kind of reactor where they're digesting them. Now, here's a fun fact that actually I've only discovered recently. cephalochordates, amphioxus, these are our closest living relatives. So these are the closest animals to vertebrates that don't have a vertebral column. So they have a notochord, so they're chordates. They're in the same phylum as us, but they have glide symmetry. So the muscles on each side of a lancelet and amphioxus are offset in exactly the same way that the segments of these other animals, filter-feeding animals, are offset, and they are filter feeders, right? So when they're feeding, they're sitting on the ocean floor, filtering bacteria. But when they want to move around, they pop up and they swim around just like an ordinary little fish. Now, here's the kicker. It turns out that you can get vertebrates to express this glide symmetry. So you can get somite formation, vertebra muscle block formation to exhibit this 90 degrees, this slight offset, just by blocking a single gene pathway. And this is an example in chickens, but it's been shown in amphibians, in mice, in fish, same deal. Single gene pathway can block the symmetry of vertebrates and take us to that asymmetric condition. So you single gene switch. And what all of the people who've done these experiments have concluded is that vertebrates have an underlying asymmetry, which we know, right? We're essentially asymmetrical creatures. Our internal organs are asymmetrical. Our brains are asymmetrical, actually. That asymmetry is fundamental in development, and it looks like it's ancestral. So then you get to the point, well, so here we have this glide symmetry, which is a specialization for this new feeding mode, for exploiting this new resource, using a new way of breaking down food. It kind of becomes a moot point whether you say it's convergent evolution or an ancestral condition, right? Because there's only one gene involved, right? So you can get this morphology out of any of these guys with a single gene mutation in multiple ways. And so it kind of becomes a moot point to say that this asymmetry is this random oddity. It's not a random oddity. It's the underlying symmetry of everything here. It just happens to be expressed only for that particular feeding mode, whenever that feeding mode of feeding using cilia sitting on the ocean floor turns up. Anyway, moving on. These things called placozoans, which animals which you've probably never heard of, are particularly interesting because they're animals that we find today. They don't have any body segmentation. They don't seem to have any morphology at all. These days, they group with, they're clearly grouped in with these bilaterally symmetric animals over here, or the eumetazoans over here. And on morphological grounds, they seem to, and behavioral grounds, they seem to quite closely resemble these Dickinsoniomorph type ancient animals. So they're a modern animal that looks like a simplified version of a Dickinsoniomorph, except that the genetic evidence is fairly clear that they're not a simplified version of a Dickinsoniomorph. It's the other way around, that Dickinsoniomorphs are some more advanced version. In other words, these things haven't evolved by simplifying this. These things have evolved. The Dickinsoniomorphs have evolved from something like this. And what's really interesting to a neuroscientist is that these things don't have any neurons or nervous system, but they do express lots of neurotransmitters and neuromodulators that you find in vertebrate brains, including the human brain. And in fact, the complement of neurotransmitters and neuromodulators that you find here is much more similar to what you find in our brain than what you find in the crunchies and the squishies, right? The insects and the mollusks, right? So, and the evidence suggests that even though these things are more closely related to us in the sense that they've diverged from us more recently, although it's back here, molecular evidence shows, these things are much more evolved. These things are much more derived and specialized. And that's particularly obvious if you look at gene expression in brain development or nervous system development in these things. So I've done a lot of work over the years on the inner ear vestibular system. If you look at the transcription factors that get expressed when the inner ear develops in vertebrates, you see the same transcription factors being expressed in the ecdysozoans in relation to their vibration sensors, their various vibration sensors, except that in those creatures, they have lots of different kinds of vibration sensors scattered all over their body. So they've got much more specialization, more transcription factors have been added into this hierarchy. So they're a much more evolved, specialized kind of creature than us. And the evidence is suggesting that, in fact, we have evolved, we are the primitive form. So when people look at a diagram like this and you see, oh, the common ancestor of a human and a jellyfish back here probably look like a jellyfish, right? That would be the normal assumption. But there's no reason to assume that, right? The common ancestor here could look like or not look like anything that's up here, right? There's no reason to assume that it's this. In fact, there's a good reason to assume that it's not that, because these creatures are specialized for feeding on larval crustaceans and vertebrates. So these things, this morphology is specialized for feeding on things like this and things like this. So it can't possibly have evolved back here before things like this evolved, right? So this is specialized. If we look at the evidence, it's suggesting that we go back and ask which of these creatures up here is most like the common ancestor of these Ediacaran creatures, you go for this one, right? Which is actually quite a close relative of us, right? So there's no reason, just apart from pride, there's no reason to think that we are not among the most primitive of animals on Earth today. And that does seem to be the case, that our brains appear to be, brains and sensory nervous systems appear to be much simpler, have a much more primitive design than these other creatures over here. We just have really big brains. That's what marks us as special. So we're going to go back and now have a look at these creatures called trichoplax that you haven't heard of. They have apparently no morphology. They're just these sheet-like creatures, just a little sandwich of epithelium. This guy is about 100 microns across. Considered to be a subtropical, tropical species until a few years ago, but we found them a couple of years ago. They're in Dunedin, they're around here. We can catch them reasonably easily in the harbor around here. Here's a video of one under a microscope. So the diameter here is about 100 microns, 10th the width of a human hair, very small. Incidentally, about the size of a single grain of sand. When those Dickinsoniomorphs are fossilized in Ediacara, they're fossilized in sandstorms. This guy is about the size of a single grain of sand, so they might have been there, but they would never appear in those fossil beds. And I could look at that for hours. We've spent hours not finding them. But here's what's interesting about trichoplax. So this, I should have put the reference on this. This is not from our lab, but this is really cool. This is a trichoplax feeding on a type of bacteria called Rhodomonas, which is red. And they have this amoeba-like movement, and you see it's breaking up the Rhodomonas underneath itself, right? So it's digesting the Rhodomonas. And as there's a kind of blue-green algae, a kind of bacteria. As those bacteria break up, as they are releasing that red stuff into the fluid, and then you see the trichoplax absorbing that fluid, eating it. This is sped up, by the way. This is, I forget, something like 20, 30 times real speed. Right, so it's attacking those bacterial algae, breaking them up by external digestion. So it's breaking them up by, so this is that the new kind of feeding mode taken into a new kind of environment where instead of like the sponges do and like our single-celled ancestors do, instead of eating bacteria and tiny food particles by phagocytosis, so an individual cell swallows a tiny food particle, these guys are putting their digestive acid and enzymes out into the environment, breaking up the cells in the environment, and then absorbing the pieces by phagocytosis and endocytosis. This is a much more efficient feeding mode than the feeding mode of sponges or our single-celled ancestors, right? This allows us basically to act as these combine harvesters going across the ocean floor and just sweeping up other organisms, single-celled organisms in large mass. There's targeting here, right? So those digestive juices are expensive. The digestive juices are only released when there's a sufficiently large cluster of prey animals or prey organisms to digest. So there's a taste map, a chemosensory map here, where if the trichoplax can taste a sufficient quantity of food, then it will release its digestive juices. Then it will stay in that location until it absorbs all of those nutrients, and then it will carry on hunting for more nutrients. So this is my point here. So that stomach is acting like a kind of brain, right? So the stomach has the chemoreceptors in it, the taste receptors. The taste receptors are mapping the world outside the organism. They're finding nutrients in the world, and then they're instructing those gland cells to release the digestive juices in the locations where there's a sufficient amount of nutrient to justify the expense of doing that, right? So this is what our brains do, right? Our brains are mapping the world and finding resources and finding things we want to go get and finding things that we want to move away from. In this case, the map is being done in indirect registration. So the map is actually sitting on top of the world that's being mapped, and it's exactly the same size as the world that's being mapped. Right? So it's a two-dimensional map of a two-dimensional world, and that's being used for making decisions about feeding, and that makes feeding efficient. That's what makes these things efficient and does help them to survive for 600 million years or so. But they do have some internal structure, surprisingly, even though they are these very simple animals. When we look at the expressions of transcription factors and the various cell surface molecules that get expressed in trichoplax, we see a kind of radial pattern. So we can see a central region here, which is the digestive region, which contains the cells that release the digestive juices and the cells that take up the digested material. Around the outside, there is this marginal zone, which doesn't contain, right? So this is the stomach, and then there's a region outside the stomach. Around that region is expressed, I don't have a picture of it here, but it's expressed one of those segmentation genes. So the genes that specify the segment boundaries in humans, the Hox genes, one of them appears around the rim of trichoplax like this. And that's a whole other story, but it's a really interesting story, what it's doing there in an animal that doesn't have any segments, but that's a talk for another day. What's interesting is that in that marginal zone, you also see this PAX-B expression. That's a transcription factor that in us, in other bilaterians, and in particular invertebrates and mammals, is a transcription factor that you see being laid down in locations where a sense organ is going to appear, right? So where your eyes are going to be, where your ears are going to be, these PAX genes get laid down saying, this is where we're going to build these different sense organs. And you go into the arthropods and you see these PAX transcription factors being laid down everywhere there's going to be a sense organ on the body surface. And yet there are no sense organs in these things. They have the transcription factors that would be a signal, were considered to be instructions for building sense organs, but now we see them in an animal that doesn't have any sense organs. They're not there. If we look at the behavior of trichoplax, these things move by, so they do have a contractile epithelium. They don't have specialized muscle cells, but their epithelium can contract. Basically, the whole body cell can contract. There are these essentially two layers of epithelium with this kind of slimy stuff in between. They, if you put them in a petri dish in a uniform environment, they will just move around at random. So they'll move as if they were particles undergoing Brownian motion on a uniform background. But if you put in a Q gradient, a chemical Q gradient, glutamate and glycine are very good attractants for these things. then they'll still undergo this random motion, but that random motion is then biased towards the source of the cue. And so they will move towards the source of that substance and start feeding on whatever's producing that substance. I think it might not be a coincidence that glutamate and glycine seem to be the primary attractants for these things that they use for finding food, because if you're a neuroscientist, of course, you know that glutamate and glycine, glutamate's the most common neurotransmitter in your brain, right? So it's a signaling molecule in your brain. It's a signaling molecule that controls the behavior of these organisms, or that modifies the behavior, causes these organisms to reward cue that they're following. As I said before, these things express lots of neuropeptides, and they have receptors for those neuropeptides. And if we put on the human neurotransmitters or mammalian vertebrate neurotransmitters and neuromodulators, they affect the behavior of trichoplax in kind of weird and not very easily understood ways. But it's clear that these things having an effect, but it's difficult to tell what the normal role of those things is, because we're just soaking these things in the neurotransmitter, so it doesn't really tell us what these things are normally doing in these animals. So it's aware that they have neurotransmitters, they have neurotransmitter, neuromodulator receptors, and that those things are clearly affecting their behavior, even though they don't have any neurons or nervous system. And this is just data showing that these things are behaving exactly like drift diffusion, acting like Brownian particles with a sense of going towards the cue source. This is looking at, so here we've just lit up, again, it's not from me, not from my lab. These people have lit up the mucus. These things have a kind of a mucus coating that they're trapping things in underneath them. And you can see that their feeding mode, as they're moving around on the bottom of the petri dish, they're stopping, and they'll stop for a while and feed. 1, 2, 3 are the places where this thing stopped and fed. So it stops and it feeds at certain points, moves around, stops, feeds, moves around, stops, feeds, which is exactly the behavior we see in these Dickinsoniids in the fossil record of South Australia, that these flat animals are moving around on the microbial mat, stopping, dissolving away the microbes underneath them, moving on, stopping. And so they're stopping because they are releasing these expensive digestive enzymes, right? So when they release those digestive enzymes, they hang around to get the nutrients rather than wandering off, right? So they get their reward before they move on and find another place to feed. Here's just a picture of trichoplax, the ventral gland cells. These pink things are the ones that secrete the acid and the enzymes. The acid and enzymes get trapped underneath the body, so they kind of stick their edges down and make a little pocket underneath themselves, and they produce acid and enzymes in the space underneath the body that breaks up any bacteria and eukaryote cells, or not all of them actually, because some bacteria are adapted to acidic environments, the acidophilic bacteria. They survive in that environment, and they actually help to break down the other blue-green algae and eukaryote cells that get digested underneath the placozoan. So this feeding mode allows placozoans to just cruise across these microbial surfaces and eat these things en masse. In the old days, in the Ediacaran period, the entire photic zone of the ocean was covered in slime, that's blue-green algae and other eukaryotes. These days, placozoan food is much harder to find, right? So you get these bacterial films on rocks in the ocean, but it's much harder way to make a living these days, but they still manage it. So in principle, if one of these things was to glide over the top of another placozoan, which would have been easy in the old days, because these things kind of, as you can see from the fossil record, they sink into the mat when they feed, right? So they're digesting the mat underneath themselves, they sink into it as they feed. Another one could come along and start eating it, and that would be, that would be kind of nice, right? Because if you want to get nutrients from making a placozoan, another placozoan is a much better source of those nutrients than blue-green algae, right? This has got all the stuff you need right there. So there's an advantage if you can feed on those guys. So there's nothing to stop one of these guys crawling over the top of another one and eating it, apart from the fact that it never happens, right? You put hundreds of these things in a petri dish and they don't crawl over the top of each other. If you have a bunch of them, they show some kind of social distancing. They'll maintain a distance from each other. They have some other behaviors, but you never see one crawling over the top of another. So it seems like they know each other's there and they can see each other from a distance. And they also have a fast escape reflex, which I know from bitter experience because I've lost a few this way, is if you bring something up near them, like a fine needle or something, before you touch them, they just curl up into a little ball and float away and you never see them again. So they have this fast escape reflex, again, despite not having any neurons. Trichoplex has an electric field, and this is, we haven't measured it, I haven't measured it, but it must be true because they're producing this proton flux, this current of acid. So acid is hydrogen ions, right? Protons, positively charged particles. So they're digesting things underneath them by producing this flux of positively charged ions. And that flux of positively charged ions is going to create an electric field around them, which is going to surround it with an electric field. And the reason I think that's interesting is because Trichoplex also seems to have an electric sense. So it has these lateral gland cells. On the apical faces of those lateral gland cells, it has voltage-gated calcium channels. Those voltage-gated calcium channels are homologous to, in fact, very closely related to the voltage-gated channels that shark electroreceptors use to detect electric fields from other animals. And as I guess you probably know, that's how sharks find food, or that's how many sharks find their food, right? So sharks can hunt in dark or turbid water by detecting weak electric fields from other animals. That electric sense that sharks have is primitive among the vertebrates. In other words, it's not something that's evolved only in sharks. It's something that seems to have been present in the very earliest vertebrates in the early Cambrian, and most of us have lost it. So we've lost our ancestral vertebrate sense. The shark electric sense is actually detecting electric fields that are due to ion flow across epithelia. So you'll often read, or if you see this on TV or something, they'll say sharks are detecting electric currents produced by muscle contraction. That's not true. The sharks are detecting electric fields that are due to ion flow across, mainly across your gut, right? So when you're in the ocean, you can stop moving, you can stop breathing, but you cannot stop that ion flow across your gut. And that's what the shark is seeing, and that's why it's going to come and kill you, right? And the sensitivity of the shark electric sense is about, or the threshold for detection is about 1/10 of the power of the thermal noise in the receptor, right? So these receptor molecules are so sensitive that they're randomly flipping open just because of thermal noise, because you're not at absolute zero. This makes them extremely sensitive to electric fields. But in fact, that level of sensitivity is pretty close to the equivalent for human hearing. So the threshold for human hearing is about 1/10 of the noise level due to thermal noise vibrations of your auditory receptors in your ear, right? So your auditory receptors are shaking around because of thermal noise. You can hear sounds that have only 1/10 of that much energy somehow. So these things have voltage-gated calcium channels, indicating that somehow they are specialized for detecting electric fields. They also have neurosecretory mechanisms. So they have the mechanisms that release neurotransmitters in neurons, right? Even though they're not neurons by any definition. They have the synaptic mechanisms for releasing neurotransmitters into the interior of these animals. So it's possible that these cells could be used to detect an approaching animal, like another placozoan or maybe a mollusk that might be something that might crawl across the top of you and eat you.
Mike, can I make a comment real quick?
Sure.
We have another thing at 1 o'clock, and I'd like to leave at least 5 minutes for questions. Is it possible for you?
Oh, what are we looking at? I've got 30 minutes on my clock. Am I going too slow? Okay, we'll move on. All right, so what should a placozoan do when what is the time, Travis?
We have about 10 minutes left.
Oh, wow, okay. All right, so we better move up. You can read about this in the next slide. So you might think that when a predator approaches, you should run away. The answer is not to do that. You should wait until the predator gets to a certain distance, and then you should run as fast as possible. So you can predict from Bayesian decision theory what a prey animal will do when a predator approaches, and placozoan should do that. It's conditional on the evidence, right? Because if you're a prey animal, you don't know where the predator is. You have to infer where the predator is from sense data. So we can take a model of these electroreceptors. So we can say, what's the probability that one of these receptors is in the open state when there's another placozoan at a certain distance? Andre or Travis can do that calculation for you. This is just to show you that we can solve that problem analytically and numerically. We can calculate what the Bayesian solution is for that problem. I'm just going to show you that a placozoan can do that calculation. So what you're going to see is a computer simulation. Here's my prey placozoan, predator placozoan. Here's an open receptor, here's a closed receptor. In the middle is the taste map that we've seen before, where it's using taste to determine whether there's something out there. And in this marginal zone, we're going to construct a taste at a distance map. So the taste at a distance map is going to be mapping things that are beyond the edge of the body. And in Bayesian theory, we call that a belief. So this is the placozoan's belief. And it's a probability distribution of predator location. And I'm going to project that belief out into the world so you can see where this placozoan believes the predator is, right? Moving along. And we're just going to do this by a reaction diffusion process. So we're going to have molecules that are released by those receptors, whose probability of release depends on that probability we showed you before. Belief molecules that are going to diffuse around. And if a belief molecule encounters a sensory molecule, that's going to catalyze a new belief molecule. So the rule is simply, if an observation coincides with a belief, you reinforce the belief, which makes kind of sense, and it actually turns out to be Bayes' rule. Moving along, here's the simulation. So here's my Bayesian calculation. Here I'm doing the calculation by numerical Bayesian inference. And this is where the Bayesian, this is where God would think the thing is, given that data. And here's this collection of belief molecules, right? So a simple reaction diffusion process is doing Bayesian inference here. Has to solve the problem of what's the probability that there's a predator within a certain range. So here's the true Bayesian calculation given that data. Here's the reaction diffusion process estimate given the same data. Matches up pretty well. How do you make that decision? Well, we can just plant a cell into that region of the map, and then we can just use that cell, put receptors on that cell, so that just measures the number of belief molecules in the environment by the number of belief molecules that bind to it, and then generates some kind of a signal. We know that there are spiking cells in this marginal zone of trichoplaques. Don't look like neurons, but they generate sodium action potentials. And I've called this an M cell because this kind of behavior, the triggering fast escape behavior, is found everywhere. So if we look at here's the true range to the predator. Here's the probability that there is a predator within the receptor field of the M cell. Here's the exact Bayesian computation in blue and the placozoan computation in brown. So this model gets pretty close to being Bayes optimal without a nervous system. So we can solve this problem as a 2D inference problem for extending a map of the world from the region of the world that's in direct contact with your body surface to a map of the world which is beyond the body surface. There's a really simple solution to that problem, and it's really simple for an animal like a placozoan to do. And in fact, the solution generalizes to, and it works because the behavior of the animal matches the behavior of the molecule. So the diffusing molecules are essentially models of placozoan behavior. I'm going to skip through this because, well, diffusion scales badly. I'll just put the numbers down the bottom. we're simulating a diffusion process here where we're releasing something here and we've got a sink over there. As we try and scale this up, it goes really, really slowly. It's slow by orders of magnitude. If we look at diffusion times for signaling molecules, nitrous oxide, which is a really small signaling molecule in the brain, glutamate, which is also quite a small signaling molecule in the brain. And you can see the time it takes to, the average time to diffuse a certain distance. When you're in the sub 100 micron range, you are below the speed at which a tricoplex moves, right? And so you could use small molecules like this. This mechanism will work in a tricoplex using small molecules like this, but it's going to fail if a tricoplex gets bigger than about this. There's kind of a hard barrier here. It cannot work at larger scales. But I just want to point out there's a real simple solution to make it work on larger scales. If I solve this problem first on a 3x3 grid, then a 6x6 grid, then a 12x12, and then I keep, as I've got that partial solution, I keep refining it, then I can solve the hard problem 50 times faster, right? So orders of magnitude faster by having this hierarchical coarse graining going on. That's a trick that numerical people use, but it means that we could scale this diffusion process up if we had some kind of, if we broke it down to into pieces and had some kind of coarse graining going on where you could rapidly send information across those pieces, telling different pieces what's going on in other parts of the solution. And if we look at neocortex is broken up into functional modules, each of which is about the size of a tricoplex. Cerebellar cortex, the same, broken up into functional modules, each of which is about the size of a tricoplex. And you can solve diffusion problems in arbitrary dimensions by linking together networks of these things in essentially a two-dimensional sheet with connections going across the sheet. So that's my bottom line. That's a little bit faster, but you can watch the presentation in slow motion. So that's what I think is going on here. The nervous system is a communication system that's linking up these chemical brain modules, which are doing Bayesian inference, solving these high-dimensional Bayesian inference problems using this hierarchical coarse graining system. What do you got? 5 minutes to ask me questions?
Thank you, Mike. How can animals perform computations without nervous systems? It's an unorthodox thought, but I think it's worth exploring. Do you have any questions for Mike in the audience? Has anybody heard of we got one?
Well, yeah, I do have a question. It's just I wondered if Mike could just go into how the coarse graining improves the speed.
The question is, how does the coarse graining affect the speed and certain method of diffusion, the slowness of diffusion?
It's because, so here we've got a 3x3. So the solution of this diffusion problem is we're just using a rule for neighbors to update each other, a diffusion rule for how much of this stuff is going to go this way and that way and that way. You with me?
Mm-hmm.
Yep.
And so you solve the 3x3 problem, it's really quick, right? Because we've just got this 3x3 grid, we solve that problem. Once we solve that 3x3 problem, we then break that 3x3 grid up into, we halve, right? We double the number of cells in each grid. And now we solve the diffusion problem on a smaller grid. You understand me? But you see, now you're already a long way towards the solution, right? So instead of, if we'd started this, what is this 6x6 grid? If we'd just done the 6x6 grid, it would have taken much longer to get to this point, right? And you can do this, right? And you can do this simultaneously. So you could simultaneously have some connections, a 3x3 grid of coarse connections across the network. So instead of the diffusion process just telling this grid here what's happening here, you send that rapidly across, right? So you have a 3x3 grid of coarse connections, and then within that you have this subdivision, and then subdivision and the hierarchical subdivision, which you can do simultaneously. In this simulation, I'm doing it sequentially, but you can do it simultaneously. With me?
Good. Any other questions? Good. Check the time. Yeah, it's 1 o'clock. Perfect timing, Mike. Can we give Mike Paulin one more round of applause, please? Thanks for your time, Mike. I'll be in touch shortly. We'll let these guys get out of here.
Okay. Okay. See you, Travis.
See ya. And we can